body is a product of different chemical reactions and processes. But what is the vital force that regulates these reactions? It is enzymes. In a cell, most chemical reactions do not occur spontaneously. Instead, they rely on enzymes, which are biological catalysts that speed up the reaction inside the body by modifying specific substances called substrates, allowing the cells to obtain the most energy resources available to them. But how exactly does a tiny enzyme accelerate a chemical reaction? Enzymes are highly selective, and a substrate binds to a specific region known as the active site. They lower the activation energy of a reaction. When the substrates bind to the active site, they form what is called the enzyme-substrate complex. As they begin to react, some of the chemical bonds and the substrate begin to weaken, causing them to link together, and the chemical reactions at the active site eventually lead to a different molecule, which is referred to as a product. Good day everyone, the focus question for today's experiment is why do enzymes react differently? We conjecture that by the end of the experiment, we will be able to observe enzyme specificity and environmental conditions that may affect how enzymes react or function when exposed inside and outside of the optimal range. Throughout the different tasks, we aim to identify enzymes found in various food sources, recognize their specific roles, and identify factors that may influence enzyme activity. The materials that will be utilized in the experiment are knife, fork, measuring spoon, stove, disposable cups, coffee or cloth filter, containers with food cover, cooking pan, cups or jars, apple, pineapple tidbits or chunks, meat tenderizer, onion, potato, fresh milk, water, cooking oil, white vinegar, hydrogen peroxide, and baking soda. For the procedures, without further ado, let us now begin.
In this experiment, I test the enzyme reaction that occurs from a fruit such as apple. The materials are peeled apple, meat tenderizer powder, measuring cup, salt, knives, and a cooking pan. First, I chop the apple into small bits. Wash the apple from a small basin. And for the tenderizer powder, I transfer 5 tablespoons into an empty cup. And mix it thoroughly. Back to the chopped apple, I added 200 ml of water and let it simmer until the apple became soft. When it's now tender and soft, I use fork to mash the fruit. Then I transfer the now puree in an empty cup and I added salt solution that I made off screen. The salt solution is 1 teaspoon of salt and a cup of water, and I added the tenderizer solution into the mix and stir it thoroughly. After that, I filtered it by using a towel, an alternative for paper filter or coffee filter. Before I made my observation, I waited for 10 minutes. The puree and filter are yellowish in color, which I assume is due to the salt. You can see a little foam forming on the surface of the filtrate.
After preparing the materials, I labeled each of the containers with control, water, oil, lemon juice, and onion. I then cut 10 cubes of apple and placed 2 cubes in each container. Then put enough water, lemon juice, oil, and onion just to cover the surface of the apple cubes. I also made a salt solution by putting 1 teaspoon of salt in a half cup of water and poured 5 teaspoons in each container. Place them all inside the refrigerator overnight. After leaving them overnight, I noticed that the control sample has the most extensive browning and the other 4 samples almost look the same in their appearance since there's nearly no change or visible browning. But if I'm going to rank them based on the barely visible differences, the control would be the first, which has the most extensive browning. The second one is the water sample, third is the onion sample, fourth would be the oil sample, and the sample with the least browning would be the apple cubes with lemon juice.
Good day everyone. So today, we are going to perform a certain task in enzymes activity experiment, which is the task 3, the bubbling veggies. But before going to the experiment proper, let us first prepare all the things and samples needed. So let's start in preparing the samples. The sample needed in the experiment are potato, onion, apple, and pineapple that should be cut into cubes. Then after preparing the cube potato, onion, apple, and pineapple, the next thing we should do is to prepare the salt solution. In doing this, let's add a half cup of water in the plastic cup and add a 1 teaspoon of salt in it. Now that we are done in preparing all the things needed in the experiment, let us now proceed to the experiment proper. So prepare a 4 plastic cup that will be used as a container in the sample. Then in each cup, add 1/4 cup of hydrogen peroxide, and after, put a cube of potato in the cup 1, cube of onion in cup 2, cube of apple in cup 3, and cube of pineapple in cup 4. Then, add immediately a 5 teaspoon of the salt solution in each cup, and observe what happened in terms of the bubbles produced. So after we finally performed the experiment, I observed that the bubbles produced in each sample varies. In cup 1, I noticed that the onion float and a little bubbles can be seen in the surface of the onion. Same is true in cup 2, wherein the apple float and a little bubbles is evident in the surface of the apple. However, in cup 3, the potato sunk and there is a continuous formation of bubbles that is rising to the surface. While in the cup 4, the pineapple sunk but there is no formation of bubbles.
Here are the materials used for this experiment. The first step in this procedure is to cut thin strips of a potato, which will be our food source in this experiment. The potato contains an enzyme called catalase, which is known for breaking hydrogen peroxide that is known to be a harmful byproduct in the process of cellular respiration. To continue with the experiment, I added 5 teaspoons of vinegar in the first cup. Prior to the experimentation proper, I prepared a baking soda solution by dissolving 1 teaspoon of baking soda in a half cup of water. Moreover, I also prepared a salt solution by dissolving 1 teaspoon of salt in a half cup of water. Second cup, I also added 5 teaspoons of baking soda solution. I added 1 strip of potato to each cup, the first and the second. The potato strips were soaked for 5 minutes. During and after the process, no visible changes can be observed as of yet. Using the third and fourth cups, I also added 5 teaspoons of hydrogen peroxide. In this case, the hydrogen peroxide will serve as a substrate for the enzyme found in the potato. Potato strip on the cup with vinegar was transferred to the third, while the potato strip on the baking soda solution was transferred to the fourth. To finish the experiment, I also added 5 teaspoons of salt solution to the third and fourth cups. Before the potato strips were added to the hydrogen peroxide, no significant changes can be observed. However, as they were added, immediate bubbles can be seen floating in the solution as a proof of reaction in the fourth cup. This reaction is caused by catalase, an enzyme within the potato. You are observing catalase breaking hydrogen peroxide into oxygen and water. The third cup, however, remains unchanged. And the difference between the two could be the result of the two reagents between the vinegar and the baking soda.
After gearing up with proper protective equipment, we start the experimentation by heating first 80 ml of apple juice in low heat for 2 minutes. After heating, transfer the heated apple juice in a separate container, then proceed by pouring 100 ml of milk stored in room temperature in both of the 2 glass containers. Next, you add 1 tablespoon of apple juice stored in room temperature into the first glass container, then pour also 1 tablespoon of heated apple juice on the second glass container. After 5 minutes, there is no significant visible and physical changes between the two solutions other than their taste became slightly sweet. But after 24 hours, the solution of milk with room temperature apple juice has formed very small amount of clumps and bubbles floating on its surface, while the solution of milk with heated apple juice, there were also clumps that had formed on its surface but is larger in amount and thicker than in the first solution.
And that concludes our experiment on enzymes. I hope that our collective results in this video presentation have contributed to your overall understanding of enzyme activity. This is Group 8 presenter, Chabita Baliton, bringing you lab experience at your homes.